So our next presenter, and the title is on the slide there, is Vett Lloyd, who's a professor in the Department of Biology at Mount Allison University. Welcome, Vett. Over to you.
All right, now that I've adjusted it for short people. Okay, so thank you guys all for showing up. I'm going to be talking about my favorite research organism here, which is the fluffy thing, but I'm also reasonably fond of humans. So let's see how this works. We'll start with the usual somewhat boring disclosures. I'm an academic, so no one actually cares about me. The research in my lab is funded by NSERC, so basic research funding. And that's about all I have to say about it. There's no commercial. I do have other grants for other projects, but I'm not talking about them here. In terms of what I am going to talk about today, the talk comes in two chunks. They're quite closely related. The first is a compare and contrast. Not only do I like dogs, I think they are absolutely phenomenal ways to use as a sentinel to see what the risk is of human Lyme disease. So I'm going to be talking about surveillance approaches, and I might as well tip my hand now and say that I'll be suggesting that using dogs as a sentinel species is a very powerful approach. The second thing I'm going to do is demonstrate why that might be the case, using New Brunswick as a case study, because we have very good high-quality tick data as well as dog seropositivity data from New Brunswick. All right. Before I go on, I'm going to mention that most of the work I'm talking about is published or in press. It's based on a canine seroprevalence study that's in press right now in the Canadian Veterinary Journal. Published work from the National Microbiology Lab, and unpublished work, which will get published soon, on our passive surveillance of ticks in New Brunswick. Okay, so Dr. Ogden gave us a really good background on how we can use tick surveillance to assess the Lyme disease risk, which is great because then I don't have to spend a huge amount of time doing that. So the picture at the far end, far left, shows some students from the labs doing active surveillance, and that's approach where you go out and you're dragging things around or poking shrubbery with little bits of flannel. You're hoping to catch the ticks that are up and about and actively questing. There's also passive tick surveillance where you get veterinarians to peel the ticks off dogs, children's and their client, and mail them into you. That's our mailed supply from one day. The drawback with both of these tick surveillance methods, as Dr. Ogden mentioned, is that they are very labor intensive. Even if you're using students or you're partnering with community groups, which is something I find a wonderful thing to do and very, very powerful way in terms of education as well as getting extra help, it's still expensive because you have to get people to different areas, and you have to then wander around in the field getting bitten by mosquitoes, black flies, and almost everything else, searching for the one tick that might or might not be questing at that particular time. And it's fairly low sensitivity because ticks' activity, sometimes they come up, sometimes they're down. It depends on the time of the day. It depends on the humidity in the air. It depends on the season, whether or not that stage is actually questing at that point. It depends on what you've treated your flags with. We determined empirically that if you rub your crag blanket over a wet, smelly dog, you get 4 times the recovery rate, which is great until you think about what happens if you travel in a car with wet, smelly blankets for a few hours to a field site. So it's not an ideal approach, and it's certainly intensive, expensive, and low sensitivity. The other end of the spectrum is human case reports. With human case reports, I don't think that there's much question that is underreported. There are many reasons why the difference, with them, we don't have to get into that. That's something for the clinicians. So I'm going to suggest that canine seroprevalence is a very nice middle ground. It's less labor intensive. There's very well-established methodology. And the beautiful thing is it's already being done. So there's actually not a whole lot to be done there. In Canada, there have only been a few studies using canine seroprevalence as a sentinel species for human risk. It's been used very extensively in the US, it's been used in Europe and Asia, and perhaps Canada should jump on board here. The reason why canine seroprevalence studies works is very simply because that's a typical family, and generally dogs live with people. They go out with people or people go out with them, as the case may be. And so you're spending time with your dog lives in the same area as you do, and for a dog to become infected requires exactly the same thing for a person to become infected. It means a tick has to find the animal, the tick has to get onto the animal and then bite and feed for long enough to transmit infection. So the mode of infection is the same, and as your dog is with you, there is that indicates that if your dog gets infected, you are at risk of infection as well. Okay, the last advantage of using canine seroprevalence studies that I'll mention is that the fact that this is already being done and the information on canine seroprevalence is already freely available online. It's the Pet Disease Report by IDEXX. They have 95% of the market share in veterinary diagnostics. So mostly this is up there. This is Ottawa. All you do is type in your postal code if you want to get really local. I thought Ottawa would be appropriate because that's where we are. And you can see the number of positive cases in your area, and that will give you a very effective way of assessing your risk. And it's quite useful in terms of public education and awareness. And again, the methodology for canine seroprevalence is very highly standardized and highly validated. Okay, so that's my dogs are great approach. Now I'm going to go through our New Brunswick study as a case study for the use of canine seroprevalence as a predictor of human Lyme disease risk. And I am using New Brunswick just because that's where we live, so that's where we have the most data. Presumably this approach would work anywhere in Canada, as there are people and dogs in pretty much everywhere in the country. Okay, so why New Brunswick? Because we have data from New Brunswick. That's our tick map with the New Brunswick divided into our 7 health districts. And each district, we've got passive tick collection data from it, and the color-coded depends on the incidence of risks. And in that case, the highest tick risk is in the south and the areas bordering Maine and Quebec, which is hardly a surprise. We also have, courtesy of the work done by Public Health Agency of Canada and Dr. Ogden's work, we have his very nice modeling. We have similar modeling using a slightly different system by one of my colleagues. So we have another source of modeling data. So superimposed on that, we can now start, seeing as how we have tick data and climate data, we can now ask what's happening with the dogs. So just to reiterate why this is a good idea, dogs live with people. They have a robust immune response to Lyme disease, so it can be detected quite readily. And their detection is detected with point-of-care commercial kits. Essentially, you take your dog into the vet. 8 minutes after they get blood out of your dog, you'll have a yes or no answer. And it's the same C6 ELISA that's the first tier of human testing. So what we did, we phoned up all the vet clinics in New Brunswick, which is the first time I realized exactly how many of them there were. And we were aiming for 100 blood samples from each of the 7 New Brunswick health clinics. We got 699 samples because our 700th dog died. It actually died of Lyme nephritis, so we decided to exclude it. We ran a SNAP4 C6 ELISA test and then immunoblotted afterwards, and then we did some other statistical stuff that I'll ignore. So the results, what's shown in the map here are the percentage of infected dogs in each of the 7 health regions. We're seeing similar results. The south is the highest and pretty much everywhere else, so we're in fact finding seropositive dogs throughout, and we had a good sample size in each. For relevance for the people in New Brunswick, this actually indicated a very rapid increase in canine seropositivity because the same survey was done 6 years ago and found less than 1%. So a lot has happened. There are a lot more infected dogs since this survey was last done. We did Western blotting. The results were generally consistent. Both tests had false positives and both had false negatives. Seeing as how the veterinary community doesn't have an established algorithm for interpreting Western blots, we basically figured this is complicated, and yes, veterinarians are right, it's too complicated, let's just forget about it. So moving onwards, a quick correlation between what we see with the ticks and what we see with the dogs and what we see with the climate models. Yes, they are quite similar. There's one point I'm going to point out, which is emphasized by the red oval up there, which is that we see along the coast a higher incidence of canine seropositivity than we would expect from the climate models. So we're picking up a lot of infected dogs in areas that you wouldn't expect to be conducive to established tick populations. This is a bird flyway. It's a major migratory route, so presumably we're picking up adventitious ticks. And I would actually argue that the ability to tick up adventitious, non-established, non-endemic populations is an advantage, because if dogs can get infected there, so can humans. Okay, so running through what this means for humans. We can go to the literature, and there's a study which looked, from Massachusetts, looked at the ratio of human Lyme cases and canine Lyme cases, and came up with the fact that there's a ratio. For every 6 infected dogs, there was 1 infected humans. I applied that to the population of New Brunswick and came up with the rather startling number that there would be over 6,000 people humans in New Brunswick infected with Lyme disease. And when you compare that to the public health number of 5 people, yeah, there's a bit of a discrepancy. So I wanted to reassess these calculations and see what we would come up with. So the reassessment approach is pretty straightforward. It actually came, I believe, originally from Robin Lindsay. And the idea is that the number of infected people is going to be the product of your proportion of infected ticks, which I called I instead of P, the proportion engorging on the host, that's shown in the second column, and then the number of tick bites. Now the problem here is that most people don't detect a tick, so there's no way of quantifying the number of tick bites until we take advantage of our friend the dog. Because when a dog gets infected, it's again a function of the number of infected ticks, the proportion engorging on the dog, that's a typo, it should say dog, not human, and the number of tick bites, and that'll give you the number of infected dogs. We know the first number, we know the second number, we know the last number, so we can work out the third number. And from that, we can then go back to humans. So with apologies to the people who have just gone through their tax returns, this is going to seem like a bunch of calculations, not unlike filling in your tax return. Go to Schedule A. I'm going to go through all of these numbers. So the proportion of infected ticks, that's actually, we've got good data on that. We've got data from the Public Health Agency of Canada, published data, our own data. In the published data, the range is from nearly 7% to nearly 16%. We used our data because it had the highest sample size at 12.3%. So it's a mid-range value, nicely between the published values. The proportion of fed ticks is actually quite easy to assess because ticks get bigger when they feed. So if you do a lovely little graph of how big your tick is relative to how long it's been feeding, you can then say how many of the ticks that were recovered from dogs had fed for more than 3 days, how many recovered from humans had fed for more than 3 days. And as a note here, I picked 3 days. I realized that ticks can be infective less than that. I decided in all these calculations, I went for the most conservative possible estimate. For any number of reasons. So our fed ratio is that in humans, we had 8% had fed for more than 3 days, or 60 on dogs. If the numbers before the slash are the numbers from the NML data, which I didn't want to choose because it was higher. So I went again for the more conservative numbers. Okay, number of tick bites. This is where it gets interesting. When I first thought about this, I figured, well, one reason we couldn't do this is because no one knows how many dogs there are in New Brunswick. Wrong again. Dogs are big business, so Ipsos Forward has, on a very regular basis, surveys the number of dogs out there so they know how much dog food they can push. So in 2014, there were apparently 111,000 some odd dogs. And they say that this is accurate plus or minus 50 dogs, which is astonishing. Anyway, 6%, with a 6% Borreliosis positivity, that means just shy of 7,000 dogs are infected. To produce 7,000 infected dogs, given a tick infection rate of 12% and 60% feeding for 3 or more days, would give you 90,000 and a bit tick bites. There are a bunch of calculations here, which I'm aware of. I didn't take them into account. We got multiple ticks for most dogs. The average is 1.3. Not all the ticks are found by dog owners by any means, and the tick infection rate is slightly inflated. All of that means that there are actually more tick bites than shown here, but again, I went for the conservative estimate. Okay, so now we know how many tick bites there are. The last thing we have to worry about is the fact that that is your furry friend, and that is you, and your furry friend is actually more attractive to a tick than you are, because your furry friend is furrier than you are. At least that's the case for most people. The other thing is generally humans bathe more frequently than dogs, so we smell better to ourselves, less so to a dog. So again, humans are less attractive to a tick than a dog is. Now, we have to calculate in the relative attractiveness of hosts in these calculations. That can be done from the literature. So we go back to the Lindenmayer study, which would give us 14%. I decided to take a direct measure because this is confounded with other variables, which is, if we took the data from the NML, the 2014 paper, they showed that they got only slightly more ticks from dogs than humans. But that's an ascertainment issue because they're getting ticks primarily from the medical system and less so from the veterinary community. In contrast, I get ticks almost exclusively from the veterinary community, rarely from humans. Those are independently submitted. However, those are really the only two ways you can test ticks in New Brunswick. You can send them to IDEXX to get tested for $150 US, but people don't do that because they can get them tested for free. So if you add that up, you end up with the total number of ticks from humans and dogs in New Brunswick, and roughly 1 in 5 bites a human versus a dog. So we do all this together, and we get an estimate of the number of tick bites. We multiply that all together, and what we end up with is a conservative infection rate of having 173 predicted human infections in New Brunswick for 2 years ago. So in many ways, this is a very nice and Canadian number. It's almost exactly between the high number and the low number, so that's lovely and Canadian and compromisey. There is a significant discrepancy. 5 to 6,000 is a big difference, but saying that instead of 5 people with Lyme disease in New Brunswick, there are 173 is still a significant discrepancy. So why the discrepancies? There are a whole host of reasons that could give it. We talked about these in detail this morning. They were well covered. Testing at an appropriate time, not the clinicians not thinking about Lyme diagnosis when someone goes into the ER or their family doc. Sensitivity of testing and so forth. And certainly that's something we can talk about. In terms of general conclusions, I would again support the idea that canine seropositivity studies using dogs as a sentinel species to assess the risk to humans is a very powerful approach and a lovely complement to the tick surveillance that's being done. It's inexpensive because it's already been done. It's standardized. It's wonderful. The national infrastructure is already in place, and it feeds beautifully into the education and community engagement initiatives that have to be happening. It is directly relevant to humans because people live with dogs, and it's able to pick up issues of adventitious ticks being infective, so it's insensitive to tick endemicity, which I think is actually an advantage. So that's it for me. Thank you.